Hi everyone, my name is Diana Wang, and today I will be presenting my poster on the potential of receptive music intervention on mild cognitive impairment. Um, I conducted this work as my thesis research as an undergrad at Harvard College with Dr. Psyche Louie at the Northeastern Mind Lab. I'll be graduating in May of 2020, so the end of this month, in a degree in neuroscience. So let's start. To begin, I wanted to first touch on the concept of music, a very brief definition, kind of the inspiration behind this project and why we wanted to do this. So, at its basic level, music is an auditory stimulus. It means a lot of things to all of us. It exists all around us, from the inflections in our voice to the traffic noise on our streets, but it all first gets processed through our auditory cortex. Um, Essentially, it is a sequence of sounds organized through time where these patterns of sounds are recognized by our brains. And this leads to a continuous generation of expectations and predictions that determine our own emotional responses and preferences for music. So already in this definition, we see that there is this emotional part of music, and um, it cannot merely just be an auditory stimulus. So we see this actually in all animal species, especially in zebra finches. They use music and birdsong for hunting and mating, but more so in humans, it's used as a medium for social interaction. It allows you to express different emotions, and more beneficially, it enhances creativity and abstract power. It really is a whole brain activity with many implications in its use. So we can harness this whole brain stimulus to treat neurological disorders such as Alzheimer's disease. So Alzheimer's disease, or from now on I'll be calling it AD, it affects 10% of those over 65 in the United States. So that's over 5 million Americans. Recently, there has been a growing interest in capturing the pre-dementia phase of cognitive dysfunction. This is more well known as mild cognitive impairment, which I'll also call MCI from here on. In 2015, it was included in the DSM-5 with its own canonical characteristics similar to those of AD, but to a milder degree. So a big issue is that of 2020, there are no FDA-approved drug therapies for MCI, and there's also an incomplete identification of target biomarkers. Thus, we turn to this paper in this presentation where we focus on a non-pharmacological solution to this issue. Here we introduce music-based interventions, or MBIs, which have become widely adopted as potential non-pharmacological therapies for dementia-related disorders. It's been shown to improve mood and stress and reduce stress. This is a shift from literature-centralized focus on mind-body interventions, which also work well, but music-based interventions are seen to be more non-invasive for older populations, especially if you're just listening to music that's familiar to you. Um, and it's also been shown that long-term familiarity for melody and lyrics are found across all dementia levels. So there can be high penetration into the Alzheimer's population. However, as I discussed in my introduction here, there is a lack of confidence in its effectiveness. There's small sample size and large intersubject variability. So we want a coherent understanding of the mechanisms of MBIs. This paper really has its foundational research done by Belfi and Louie of 2019. In that paper, they built a neuroanatomical model of pleasure during music listening, which involves the auditory and reward networks. And it has been said that for these networks, if they are intact, it's necessary for successful music therapy because it targets all the emotional areas and how you respond to music. And I just wanted to quickly say that this study really focuses on intrinsic connectivity of your brain networks. We are obviously interested in how actively listening to music in MRIs can affect your brain regions, but this research serves as pilot data for the future stages of the project. And so by looking at just your baseline functional connectivity, we can then have sufficient confidence that our music therapy sessions will work if our MCI populations do have preserved connectivity between the auditory and reward networks. So again, like I said, this research really is the pilot data for further research. So here we wanted to look at the intrinsic activity, where in further stages, our different lab members will be recruiting real MCI and AD patients to run music therapy sessions on and do MRI studies on. So we used open source data from the Alzheimer's Disease Neuroimaging Initiative, or ADNI. This is a really great free online database that has thousands and thousands of brain scans on them, and researchers from all around the world can upload their brain scans from their research and upload them according to a certain standard, so we know we are getting clean data. So we were able to cull 105 patients. 47 were in the control group, or CN, 47 were in the MCI group, and then 11 were from the AD group. And the small sample size for AD is due to noise artifacts and motion in the brain scanner. So we had to kind of take those out. So to begin, we had to define our regions of interest. So using past literature, including Belfi and Louis, we defined the auditory network with 18 ROIs, regions of interest, and a similar reward valuation network with an additional 18 ROIs. So all of these ROIs were taken from the CON default atlas. CON is a great toolbox that we can use in MATLAB to analyze brain scans, to pre-process them, and to run all the analyses. So we chose this atlas because it had the most number of ROIs and provided us the most power in future analyses. So we see the ROIs here all superimposed on each other. So first, we looked at seed-based connectivity, where we defined each ROI, so the auditory network as one, and then the reward evaluation network as another ROI, and looked at their whole brain connectivity patterns and between group comparisons. So in our results here, I'm going to zoom in a tiny bit. Great. We have reward areas in red and auditory areas in blue, and purple is where they overlap. So for the auditory network seed, which we see first in the blue, all groups showed highly significant connectivity to auditory areas, such as the superior, medial, and interior temporal gyrus. And this was all at the P is less than 0.05 FW corrected level. We saw that the control and MCI groups had significant functional connectivity in the parietal, occipital, and frontal lobes, while the AD group had less functional connectivity than the other two groups. Between group comparisons, which we see at the very bottom, with control group being higher than the AD group, showed a higher functional connectivity in the precuneus for the control group. And this is also really exciting to see because it shows that control and MCI groups are not significantly different, showing preservation. For the reward network in red, we also similarly see highly significant connectivity to reward areas, as would be expected. We see, again, the pattern of control and MCI groups having higher functional connectivity in the frontal, parietal, and occipital lobes. And then between group comparisons showed higher functional connectivity in 6 different regions across the parietal lobe. Moving over to, let me move my screen. Great, moving over to ROI comparisons. Let me zoom in more, so you can see better. You can zoom in also later on your own with the PDF version to see clearer. But essentially here, we made heat maps of brain connectomes between the 3 groups. So we have all 36 ROIs listed on each of the axes, and then each of the boxes correlate to the R correlation value between 2 different ROIs to find. So essentially, without going too into detail, if we look at the within network connectivity, so the auditory to auditory and reward to reward, which we see in the bottom right and the top left squares, we essentially see preservation of connections between the CN and the MCI group compared to the AD group. Especially one good thing to note is that in the reward network, we see actually preservation of the caudate throughout all three levels of cognitive dysfunction. Um, Next, we see the between network activity in the top right and the bottom left. First, it's just already obvious to see that there is less between network activity overall compared to within network. But what is notable to see, and in data that shows the MCI group actually has a higher number of stronger connections between compared to the CN group. So there seems to be this preservation not only within networks in the MCI brain, but also between the auditory and reward. Lastly, we used some graph theory measures to look at connectivity. So we graph theory, essentially, I'm not going to go too into detail, but it is a mathematical study of systems of interacting elements where elements are modeled as nodes and then connections are modeled as edges. So in our case, our nodes were each of the 36 ROIs, where the edges were a statistical relationship. So a correlation between time series as this is functional connectivity. So we found that in our network measures of betweenness centrality, degrees, and strengths, these three parameters showed main effects of groups at a correlation threshold of 0.2, which is just a well-accepted number of threshold to look at for the correlation value. So in betweenness centrality, which is the number of shortest paths from one node to another that contains a given node, so it's how used an ROI is in pathways. We see highest levels in the control group followed by MCI and then AD. There was no significant difference between control and MCI groups, but there was a significant difference between both of those groups compared to the AD group. Similarly, in degrees, we see a significant effect as well, but here the pattern is to switch so that the MCI group is actually on top with the highest levels. This happens in strength as well. And degrees is the number of nodes significantly correlated to a given node. So it's just how many actually things are eventually get correlated to that ROI. Whereas strength is the sum of the coefficients, correlation coefficients. So it's directly related to degrees. To summarize, again, for the graph theory analyses, we see that the MCI group is higher than the control and the AD groups in degrees and strengths, while indistinguishable from the CN group in betweenness centrality. Likewise, the AD group is all indistinguishable from the others in clustering local efficiency. So moving on to just conclusions. Here we defined a auditory and reward network based on previous studies and characterized their intrinsic functional activity using resting state fMRI. We see a decrease in functional connectivity within and between the two systems in 80 individuals. And one last thing to really note that's really interesting is that we see an overlap in seed-based connectivity patterns from the two networks that we defined earlier, centering on the anterior insula. And the anterior insula is a really important region in the salience network, and it's posited as this hub of kind of switching between the default mode network and executive control. And it's been implicated in Dolphin-Louis in her neuroanatomical model, where it's the centralized ROI that connects the two regions, or the two networks. And it's also implicated in lesion mapping studies, showing that it's a really great ROI and implicated for music processing for emotions. So yeah, just wanted to say thank you to the rest of the lab members. Psyche Louis, Alex Belden, Roger Burton Patel, Valerie Gautama, and Grace Wilson, who are also all currently carrying out our later stages of this project where we are recruiting our own patients. Also, thank you to Manoj Basin at Emory, who is conducting the blood genomics portion of this test. Each individual that comes in will undergo a blood draw, and we'll look at inflammatory markers of stress. For Maya Geddes at Brigham's Women's and MIT, acting as my supervisor for my thesis, and then also the great Suzanne Hamster at the Berklee School of Music for conducting all of the music therapy sessions. So thank you. Yeah, I look forward to your questions and discussing there.